While the gel is polymerizing, I like to prepare my samples. So I do this kind of side by side. You saw the tube there, that's called Lamley buffer. We'll talk in class about what's in it. It is labeled a 2x, but it is actually a 1x. I just use that same tube to do my dilutions. And then I add the appropriate amount of Lamley buffer to my cell pellets. And then what I'm going to create here is a whole cell lysate. Basically, I'm just taking this bacterial cell pellet and lysing it in the Lamley buffer. And so the Lamley buffer, as I just kind of mentioned, lyses the cells, but also prepares the proteins in that sample such that it can run in through our SDS-PAGE gel. And once that's done for all, I boil for 5 minutes. After boiling, sometimes what happens is the labels come off, so it's probably a good idea to relabel right away. I didn't, so that caused me some troubles. When you're ready, you need to gather your materials. We're going to need a power supply because, of course, we're going to use electrophoresis, electric charge. We're going to need those 2 gels that you made. We need this gel tank plus this gel holder and clamp thing. I don't exactly know what they're called. We need some running buffer. Used running buffer is just fine. We will reuse it, as we'll see later. And we need some gel loading tips, like the one that you saw when we rinsed out our our wells. But again, in the event that we don't have gel loading tips, you can use LTS tips. They don't work as well because they don't reach quite as far. And in the pinch, I've used those yellow tips, which work terribly. But again, it does get the sample into the wells. Now that you're ready to load your gel, I just want to show you how everything goes together. The clamp gasket thing, sorry, I don't know what it's called. It just sits in that chamber there. I'm just kidding. I did that on purpose. So be careful. It does slide out. So usually I'll put it on the table or just make sure that your hand is underneath and braced. It sits in that little notch, small glass plate facing the inside, okay? And then make sure that when you place it in there, it's able to close on the gasket, small glass plate, right? And just like that, nice and snug. And then you can place it into the larger holder. So you want 2 things. Make sure that it's on the bottom. Place it in a large holder. It is a lot easier than it looks. And then make sure that it sits on the bottom and then clamp it closed. Once that's done, place it into the gel tank. And then you want to fill the chamber with the running buffer, right? And so notice that our small glass plates are on the inside. We're actually going to place our samples inside. So the current's going to run from the inside and then to the outside. And so I like to pour the inside, fill the inside chamber first, look for any leaks, it's all good, then I'll fill the rest of the tank. No leaks, so when I fill the tank, I don't know why I do this, I keep filling the middle chamber and I just let it waterfall down. And then I fill it about this much, but then I decided I'm gonna fill it a little bit more. Not sure why I decided that. I just decided that. So kind of at the top of those clamps, and that's sufficient. And we're ready to load. And if you recall, you remember this? I showed this part in our how to pour an SDS-PAGE gel video. Well, here we are now in present time. By adding those little lines, it helps you see the wells because they're really hard to see right now, right? But we do have a little trick. There's a little guide here. Yeah, our beginner gel loading guide thing. We just place that in there, and then that provides channels for you to load your samples, right? And so 10 lanes is all we have, right? Including a ladder. So when we load, simply use those channels and slip the gel loading tip in between the glass plates, and that's very important. And usually I aim for the middle of the channel. All right, so middle of the channel, and just got to make sure. Usually what I'll do is I'll kind of poke in there, and then you'll feel whether you're in between. Sometimes you'll hit the glass, or sometimes as I push, I go, whoa, this is going down way too far. I missed the glass plate, right? But just make sure that you can put your tip in and then just add your sample. Nice and slow. And I think I did mention that in the event you don't have gel loading tips, you can use LTS tips. So it doesn't work quite as well because even though the tips are nice and thin, it does allow you to get in between the glass plates. It doesn't allow you to get very far. So when you pipette it, just make sure you nice and slow and then just let the weight of the sample buffer kind of bring it down. And here's a little trick that I do. Because both of the gels look the same, I put a little black dot on the top, just so that I know which gel is which, gel 1 and gel 2. You don't need it because the clamps will help you with orientation, but I sometimes forget, so the black dot certainly helps me ensure I know which gel is which. And so the last sample is always that I load is the ladder, one on each side. And we are ready to run. Now I pulled a total noob move. I forgot to take out the loading guide thing, but that's okay. As you will see, it works totally fine. So get my power supply out. We are gonna run at constant volts. Lid goes on, red to red, black to black. Make sure it's nice and snug. And then plug it in, red to red, black to black. Turn it on, and then, excuse me while I fight with the cables a little bit. Make sure that you're on constant volts, and we are going to run at 90 volts. Hit the running man, running person, and we're going to run it for about 110 minutes or so. You're going to start to see the blue dye move through the gel, and I like to let that blue dye run below that green gasket line. And a couple updates. This is what the gel looks like at 35 minutes, at 95 minutes, and it's right around 103 minutes. I decided that's far enough. I think I could have let it run a little bit longer, but I felt that was sufficient. Oops, that's when I realized. Forgot that. And then carefully pour the buffer into the tank, trying not to make a mess. And I think I did a pretty good job of that. Yay me. And so now you got to be a little bit careful with which gel you're working with and the orientations of the gel, right? And so there's that little black mark. I don't know if you can see it. Just be careful when you take this out because it's going to fan open and sometimes it'll fall and break, right? So just be aware of that. And then in this case, remember the small plate is on the inside. So my lane 1 is on the left with the small plate on the inside. And then because of the fact I put the ladder, you can actually see the ladder. It's a pre-stained ladder. And so that tells me what is lane 1, right? And then so when you peel the two glass plates apart, the gel is going to stick to whatever it's going to stick to. Now here's a note. Do you see how there's this little gel prying tool? See how janky that thing is? I don't know if you noticed how jagged it was. Maybe I'll point it out with an arrow. It did not work great. But anyways, so you peel it apart, let the gel stick to the plate that it wants to, right? And I pointed to the, in this case, my lane 1 is to the right. Because remember, the small plate's on the inside. I peel off the wells and I put that onto the other glass plate, whatever that one may be. But again, because this thing was so janky, I wasn't able to do it very well. I will redeem myself later, I promise. And then I always notch the top left of the gel. This, again, it wasn't a clean cut, but I always like to notch the top left. That helps me orient the gel because once it's floating around, it's impossible to tell. Also notice, by the way, I still could have let the gel run a little bit longer. There's still a lot of space for the dye to have moved, the proteins to have moved through, right? But I stopped where I stopped. Finally, to get it off, you stick your little gel prying tool underneath, and oh no, it's ripping because of that stupid tool. I'm gonna blame the tool. But that's okay. That's a part of the gel. I didn't need too much. Get your tool underneath. At this point, I really should have switched, but I was stubborn. I thought, I can do it. Get your tool underneath the gel. I like to lift one whole side and then let the gel fall off and into, this is simply distilled water. So I just I like to do a rinse, wash off any SDS that's left over, and then I just felt shame afterwards for such a terrible job. So to fix that, I got myself a new gel cutting tool, one with a much better edge. Repeat the process. This time the gel stuck to the small plate, so that means my lane 1 is on the left. Peeled much nicer. I notched the top left as always, and in this case I notched the bottom right to indicate that this is gel number 2. Into distilled water for a quick rinse, and then now I have 2 gels ready for either staining or Western transfer, depending on what you need to do.